people want to ask questions, although, you know, if you want to come see me, I might try and push it a little bit closer so I can go home. But I'm still happy if you can't meet me till 4:30, that's fine. So come tell me if you want help or you're working on your lab report or something like that. So, office hours. One to 5 p.m. today. I will not have, we won't hold the wet Thursday study session as you might expect, okay? Because we'll all either be recovering from Thanksgiving dinner or out doing the sales, right? We'll be in line at Walmart. Be throwing elbows at Walmart, I guess. Maybe it's all over at that point, isn't it? Doesn't Walmart have their, you know, rush and they open the doors like at 1 in the afternoon? I don't know, it just seemed like they always keep moving it back and back where that rush is, and I decided to partake in it, which I don't anymore. I felt like it was ruining my Thanksgiving. So anyway, all right, so let's see. Questions on the class, things we're doing. Okay. All right, let's go ahead. What we're going to do is we're still working on carbohydrates, and I just want to remind you where we left off. We're not actually going to use this today, but remember we left off talking about the peptidoglycan layer, which is a layer around, it's part of the cell wall around some bacteria that helps protect them. It's a target of lots of antibiotics. In particular, we were talking about the penicillin derivatives, okay? But we're talking about carbohydrates, and it's made up of long chains of a repeating dimer of a carbohydrate. And then that repeating dimer is cross-linked by proteins. So we're going to talk about another type of repeating dimer. Now we're going to start talking about glycosaminoglycans. Okay, what these are they are polymers of repeating dimers. Okay, where the monomer is, it's a monosaccharide, right? So let's say dimers, so we'll say disaccharides. They tend, like the one below is heparin, which you probably have heard of, but they tend to be very highly negatively charged. Okay, and it's in this set of molecules that you see some of the highest molecules that have the highest negative charge in nature. So these you get examples of those sorts of things here. And if I have a polymer and it's all negatively charged, what do you think the shape's going to be? Not a helix. A helix is a somewhat compressed form, right? Wound up. Think about a phone cord, right? Guess some phone cords, right? One back there. If I take that phone cord, can I make it less compressed? I can stretch it out, right? If I got a whole bunch of negative charges, they want to get as far away from each other as possible. So they're going to tend to be linear in terms of their shape. Okay, so here's an example of heparin. What does heparin do? Anybody know what heparin's used for? Stopping blood clotting. So heparin is a blood thinner. And what heparin does is it activates antithrombin. I'll show you in a second what that means. So essentially, this is heparin is a repeating, it's this disaccharide. See all the negative charges on it? And that disaccharide, we get 15 to 90 of these repeating. And they take a certain fraction, they'll inject that in the hospital. What they like about this sort of thing is they inject it, they get their effect, and then they can stop injecting it and you stop getting the effect, right? So in the hospital, one of the things they like about their blood thinners is being able to stop a blood thinner. Okay, so that's why if you go to the hospital and they give you something as a blood thinner, um, it might be heparin temporarily. If it's a little bit longer, they give give you coumadins, so it takes down your vitamin K. They can actually counteract that. How do they counteract that? You've got coumadins in your system, so that's like the warfarin. Based on what we know about that mechanism, if I was a physician and I've got that on that as a blood thinner, and suddenly I need to reverse it, what would I do?
Wasn't it like we talked about biochemistry?
Good. Let's look at it really quick, if I can find it. Vitamins, right? So we're in fats for that. We need to go all the way back to fats. Here, maybe it was here. Do I see vitamin E down there? Yes, vitamin E. Here we go. Okay. So this is what warfarin does. So warfarin is a coumadin. All right, warfarin is a coumadin, and so what it does is vitamin K is involved in this cycle that takes some of our the blood clotting factors, and it double carboxylates it. So it's involved in the enzyme it's the enzyme cofactor that carries this out. And so vitamin K is going to come around this way. It's going to get oxidized, and it gets re-reduced. And warfarin stops this cycle. So vitamin K would get used once, and then it'd be done. Does that make sense? Okay. So how do you reverse this? I have to instantaneously reverse this in somebody. They're bleeding out. What do we do with babies when they're born? We think this might happen.
Inject vitamin.
We give them vitamin K, right? You just inject more, but you inject a big dose of vitamin K into the person, and you've overcome this effect, right? At least temporarily. So this is the reason why they would like to give you a coumadin in the hospital as opposed to, say, Xarelto, which is something you would take once a day. They can't reverse that Xarelto. Okay. So if we look at the blood clotting cascade, well, we'll come down here for the blood clotting cascade. Then we'll go back up to feet. Awesome. Push the other one down. Okay, so if we look at the blood clotting cascade here, this is the same cascade I always show you, it just has different annotations on it. But for the blood clotting cascade, Xarelto is going to, and there's another one, I don't remember the name, but it's going to inhibit factor XA. Okay, factor 10A, I should say. And so if you've got this inhibitor, you can't easily reverse it. You can't. 10A is a protein. You can't easily get a bunch of 10A and inject it into somebody. You could, but vitamin K is much easier. It's much less complex. I don't know if you'd react to it, how that would work exactly. So they like doing that sort of thing. Okay, so feet. Yeah, exactly. What's going on with these feet?
The blood flow is coming.
The blood flow is different from one to the other. In fact, we've got a clot in this leg, and so we're not getting good blood flow as compared to that leg. Okay? So you worry about blood clots and you want to have these ways like heparin is an example of something they would give you to hopefully keep the blood from clotting over here, right? It wouldn't necessarily break up the clot, but it would keep you from getting more clot, hopefully. All right, and I talked about it being an antithrombin activator. So what we have is you've got a bunch of things going on here in our blood clotting cascade, which is in black, and then there's a protein called antithrombin that will come in and it will inhibit the activation of factor 10 to make 10A, and it will also directly inactivate thrombin. All right, and so these you need to go down here and get this is where your clot's down here. Okay. So heparin is going to activate the antithrombin. But what is the molecular mechanism for that, Dr. Johan, you say? So here's an example of it. And you'll see that with these. So remember, your glycosaminoglycan is So this is heparin sulfate here. And glycosaminoglycans are these very negatively charged species, right? And they can attract proteins. And what happens with this is if you have the heparin around, this is antithrombin right there. It says it in the caption below. Antithrombin will bind to the glycosaminoglycan and it'll change shape. And then it can recruit factor 10A. Okay, so it gets a shape that can bind 10A. And if 10A is not around, let's scoot back up. If 10A isn't around here, you're not going to get any thrombin, and you can't get a blood clot. You see how the heparin helps? What's going on with the heparin? Questions?
So like red lines that just stop, that's like saying it's blocking.
That's an inhibition. So that's
Okay.
when you have a pathway near one of these red lines, or not red line, but just
A line that just
a
stops.
line that stops. That means that it's inhibiting. And then if you have like an arrow, then you're talking about it activating. Okay. Another way is that so you saw that there that not only did heparin activate or did antithrombin inhibit factor A, 10A from making thrombin, right? But it also inhibits thrombin directly, and here's an example of that. So we've got our new shape for antithrombin here, and that can actually bind to thrombin, and thrombin also has a nice place to bind to the heparin as well. So now we pull the thrombin out, any thrombin that's there, you can pull that out. This is a nice molecule that you can use to kind of stop blood clotting from, if you've got stuff clotting, from you getting more clotting from it.
Can you show those like next to each other? Like
yes,
difference?
I can do that. Let me scoot up really quick.
Okay.
So what this is taught, what's going on here is we're seeing the mechanism for this, or part 2 mechanism is for this inhibition, and then a mechanism for this inhibition right here. And so this is 2 ways that essentially you're pulling things out of solution onto the heparin through the antithrombin.
Is that how it, like the antithrombin works in general? Like, because it's not just with heparin?
I'm not sure, but I'm going to hypothesize yes, or I'm going to speculate yes, because I'm about to go on and talk to you about, so heparin is kind of, it is natural, but it's not really, they don't really know what it's really for in general, and maybe it's some sort of blood, you would think it's some sort of kind of overall blood clotting manipulation, but essentially when they give you thrombin, they essentially extract thrombin from pigs. Not thrombin, the heparin from pigs and isolate it. They don't synthesize it or anything. Okay, but what we have is we have a lot of these sorts of things. Like here is heparin sulfate. So up above I was talking about heparin, but actually it says heparin sulfate. Heparin sulfate is actually on the surface of cells, and the way that happens is we have a molecule called a proteoglycan. Okay, and a proteoglycan is a protein with glycosaminoglycans stuck to it. So it has a protein anchor. So that's structural. And then it has one or more glycosaminoglycans attached. And so heparin does this recruitment, but it's really mimicking this natural molecule heparin sulfate that's part of this proteoglycan. So cells, various cells, will have these proteoglycans on them with molecules hanging off, with glycosaminoglycans hanging off for function. All right, so blood clotting, and so actually before I go on, So there's going to be two classes here. There's going to be proteoglycan, okay? And I'm about to talk about glycoproteins, right? They both have sugar and protein in the name. People often get them confused. You need to keep them straight in general. Okay, so the most important thing here is that you can tell me the difference between a proteoglycan and a glycoprotein. Okay? I haven't told you what a glycoprotein is yet. But proteoglycans, you've got a protein anchor that's structural. You have a one or more glycosaminoglycans attached, and that's the function. Okay, those have some sort of function. Generally, it's to collect things and either inactivate them or activate them. And so here's another example of that. So you can imagine if you're a cell and you want energy, right? You could get energy from fat molecules. Okay, so you have these well, you have your cholesterol molecules, which are macromolecules of things put together. Okay, it's multiple molecules together. But you have these things floating around the blood, and you have these lipoprotein lipases. So a lipase is something that breaks down a triacylglycerol into its component fatty acids. So in other words, the reaction that's going to be catalyzed here. Oh, I didn't leave myself much room. Okay, so I have these. And then you can add water, 3 waters across those bonds, and then pretend these are all the same thing, 3 of those, plus our remaining glycerol. Like that. Okay? And that reaction is catalyzed by these lipoprotein lipases. But you could imagine that you don't want that to happen just anywhere in the blood, right? You don't want it to happen in the middle of a blood vessel, especially because, you know, these lipids are, the fatty acids would stick to the walls, right? They've got a fair amount of hydrophobic character. You want this sort of reaction to happen so that the cell can then take these up. Let's talk about cell absorbs these. You'd want the cell that needs the fats to have this reaction happening right outside of it. And so a way for that to happen is instead of having these lipoprotein lipases floating around the blood at whatever concentration you need, you can have them concentrated on the surfaces of cells. Okay, so this is a way these are attracted to, let's see, does it tell us what glycosaminoglycan this is? Okay, so it's heparan sulfate again. So we've got heparan sulfate out here, and our lipoprotein lipases are going to stick to it, and so that will help the cell get the fatty acids it needs. Questions on that? So that's another example of how this might be used that isn't related to blood clotting. Okay, that's proteoglycans. Well, almost. So when we think about, we sort of in this class, you maybe in another class had built up a more complex view of the cell membrane, but the outside surface of the cell is really complex. We've looked at it in various ways, but if you think about this membrane, you've got all sorts of things in it and sticking out of it. And so here you can imagine we have this network of we've got collagen out here, which we haven't talked about all that much, but we've also got these proteoglycans. So this is, you see, this is a protein string right here with all these little glycosaminoglycans coming off. So you can imagine the outside of the surface is this very kind of fuzzy area. And what's its charge? It's a fuzzy negative charge. Okay? And so proteins that you would like to associate with cells are going to be positively charged, right? They're in your blood, they're positively charged, going to attract and kind of stick on cells as opposed to floating around everywhere. Yes?
So heparan sulfate is a proteoglycan.
Heparin sulfate is a
Oh,
glycosaminoglycan, but it can be part of a proteoglycan.
is heparin the drug and heparin sulfate the same or different?
Heparin sulfate is kind of the natural one that's on cells, and heparin is the drug. So they are different, and you wouldn't say that heparin is attached to the protein, although I'd probably just let that go. But just to be correct, heparin is a drug. It's just the glycosaminoglycan. It does not include the protein part, which would make it a proteoglycan. Okay? But your main job over all this, these are examples of these. I have to look at the study guides. I may ask you for an example of one of them. Understand the difference between a proteoglycan and a glycoprotein. All right, let's go ahead and introduce glycoproteins. Glycoproteins. Glycoproteins are essentially proteins with carbohydrates attached. Is this big enough for people to see? Okay. Glycoproteins are proteins with oligosaccharides attached. All right, I'm going to go ahead. I'll define oligosaccharide in a minute, but I'll actually write it in a minute. I'll tell you right now. These are like 20 or less sugars stuck together, monosaccharides stuck together, and they're highly branched. They're information rich. Their job is to communicate information. So the protein has function, okay? And the oligosaccharide is an information-rich tag. So for example, the oligosaccharide might signal, hey, take this outside of the cell. Okay? I'm going to give you a couple other rules as well here. But before I do that, let's quick define oligosaccharides or write it down. Okay. These are going to be short, and when I say short, I generally mean less than 20. Short or carbohydrate polymers that are highly branched and often very diverse in terms of what sugars they contain. Okay? So an example of one that is not super highly branched, we've already talked about this. I'm going to bring it up again, though, because I want to introduce an interesting way that we see this in medicine, is our blood clotting groups, right? Not blood clotting, our blood typing groups, right? So your blood type groups, we'd have a red blood cell here. That red blood cell has, if you're O, you have this shape. If you're A, you have this shape. If you're B, you have this shape, okay? Not super highly branched yet. I'll show you some more highly branched ones, but very diverse in terms of, the type of sugar. This isn't a repeating disaccharide. It's not all glucose. Got a bunch of diversity in terms of this. And so what we would say is that this right here, actually I don't have a protein attached in this case. Imagine we have a protein attached. Call that a protein. You actually end up with these directly attached to proteins as well. But what we've got here is this is an oligosaccharide. Okay, so you have an oligosaccharide there. And this is actually important in terms, and what can happen is we have a protein, or we have a name for proteins that bind to these, and they're called lectins. Bunch of vocabulary today. And they're proteins that bind very specifically to carbohydrates. All right, and why I'm using this example in terms of with lectins is here we have our oligosaccharides, and there's an example with the blood typing group that's actually related to ulcers. So does anybody, this is an ulcer, does anybody know what all those little black things are? Anybody know what causes ulcers?
No,
Not,
no,
not E. coli.
pylori.
H. pylori, right, but bacterium does, right? So ulcers, many ulcers, let's say, and this represents I guess it's been 30 years now. This represents a change in thinking. Right when I was in grad school, they were really figuring this out. And the guy that figured this out, I should know his name, but the guy who figured this out, what he did is nobody would believe him. So he was a physician. He said, no, ulcers aren't caused by stress and too much stomach acid. I mean, mostly they're caused by infections of this bacterium. And nobody believed him. And so what he did was he actually grew up a culture of this and drank it, and immediately got a really bad ulcer. And then he cured it with antibiotics. Turned around and cured it with antibiotics. So did the mad scientist thing. So many ulcers are caused by bacterium called H. pylori. Okay, and that's what's pictured over here. Okay, this bacterium has a lectin on its surface. Remember, a lectin is a protein that specifically binds an oligosaccharide. Okay, it's a lectin on their surface that specifically binds the blood type O antigen. So what does that mean if you are blood type O? What was that? You're more prone to having ulcers, right? Not only are you the universal donor, which sucks for you, but it's good for everybody else, but you're also more prone to getting ulcers. Okay, questions? All right, so that's an example of a lectin. Let me give you another example of where we have an oligosaccharide involved, and also we have lectins involved here as well. But let's talk about, so we've talked about these oligosaccharides, so they're a place for bacteria to bind. That's not something good. They're responsible for your blood groups. They can work as molecular timers. They can signal if something needs to go outside the cell. Like all antibodies have these tags that say, "Hey, export this." But so here are examples of various oligosaccharides. This one isn't very big, so it can't be branched, but you can see a bunch of the branchings over here, right? This is an example of a tag, an oligosaccharide as a molecular timer. Okay, so, and when we look at this particular oligosaccharide, I'm going to be talking about sialic acids. Sialic acids are, this is our structure, and those are the sialic acids on there. Okay, in terms of our discussion. So, your proteins in your blood, depending on what they are, they'll last a certain amount of time, right? Your body makes proteins, it dumps it out in your blood, but they run into things, right? They run into things. There's oxidizing agents in your blood, right? They could encounter oxygen and have some issues. So these proteins aren't going to last forever. And so after a protein gets old, it needs to be pulled out of the blood. Well, one way that happens is proteins that are in your blood get this oligosaccharide attached. And then as it's floating around in the blood, these sialic acids can get randomly chopped off by enzymes called sialidases. Okay, so we've got this stuff floating around. You've got any protein, antibody, let's say, floating around your blood. It's floating around for a certain amount of time. It floats around for a few days, runs into a sialidase. That sialidase pulls this one off. Okay, runs around for a little bit longer, then this one gets chopped off. Then finally runs around a little longer, then this one gets chopped off. Random sorts of events. Okay? Once all three are chopped off, the liver recognizes this shape through a lectin, right? The liver has a lectin that recognizes this shape, and it pulls that protein out of the blood and breaks it down. Okay, so it's a molecular timer. Now, it could and so proteins will have some sort of half-life based on their probability of running into a sialidase and having these three chopped off. Does that make sense how it works? So this is kind of a judge of how old something is. So let me go ahead and write that down. So proteins in your blood have oligosaccharides attached that function as molecular timers. Okay, these oligosaccharides have sialidases on the ends, and those would be probably I'm not going to say it's a reducing or non-reducing end because these things are so complex. They don't always hook together, reducing end and not, or in the non-reducing end direction. They can hook to any of those sugars. It gives you a lot of really complex shapes. So there's a lot of information that can be communicated here in terms of the shape. You have lots of different shapes, lots of different information. Okay, so but these oligosaccharides have sialic acids on the end. That can be removed by an enzyme called sialidase. Right, the longer these are in your blood, the more likely the sialidase is to be removed. All right, once they're all removed, the liver will recognize them through a lectin and pull them out of the blood. All right, questions on that? So that's an example. Once again, we've been talking about glycoproteins, right? The protein in this case, we didn't even talk about the function. It could be anything. But let's say it's an antibody. This is a molecular timer sitting on an antibody that's what as it floats around in your blood. Questions?
I thought there was the glycoproteins were the ones that where the protein did the function, and it was it is.
Yes. Got it? So the antibody, in the case of an antibody, the antibody binds to things, signals the immune system. This particular sugar is on there signaling that how long it's been in the blood. It may have another oligosaccharide that told the cell originally to, hey, let's export this outside of the cell. Okay. Other questions before we're going to move on? We're going to change topics here. We're going to start talking about metabolism, sort of, and energy usage. Okay. So living things transform energy. I know they've talked to you about this in biology courses, right? So living things transform energy from one form to another. And most of the energy on Earth that living things use comes from the sun, right? I don't know, is it wrong for me to, do you capitalize Earth? Maybe you don't.
Earth is capitalized.
Earth
You're
is?
talking about like dirt.
No, I'm talking about Earth,
A
planet.
that it's capitalized.
Do I capitalize the sun too then?
No.
No, it seems wrong. Okay, all right. So capitalize, but so most of the energy on Earth comes from the sun, and we have living organisms like plants. Okay, plants are going to take in carbon dioxide. They're going to take energy from the sun, and they're going to turn it into organic products, and they're going to release oxygen. And then we have heterotrophs, okay, animals that are going to come in and they're going to eat plants. They're going to take those organic products and they're going to break them down into carbon dioxide again that can then be used by the plants. We have the sun fueling this cycle. And we're losing energy along this cycle at all places as heat. Right. We're always going to lose it as heat. So we can think about this. Let's talk just briefly in a little more detail about the types of energy we have here. So we have light energy that comes in from the sun. And plants are going to do photosynthesis, right? And they're going to turn that into what kind of energy? Yeah, we're going to get chemical energy. Right? And we're going to lose a little bit of heat here. And then animals will come and they'll eat plants, right? Okay. So we're going to have, we want to put a process here. Animals will eat plants. Plants will eat plants. Why do plants turn that into chemical energy, by the way? Why don't they just use the energy from the sun to grow directly? Just use it to make ATP directly. Certainly, that's certainly something that can evolve.
For storage?
For storage, because the sun goes down, right? Okay, so the plant has to make it through the night, minimally. So you're going to need to be able to store that energy. So this is chemical energy. We could think about this being storage as starch, maybe. I mean, more basic, it's going to be glucose. So animals can go ahead and they'll eat that energy, and then so well, plants can use that energy, right? And they're going to take that energy in the form of starch or breaking it down to make glucose, and they can use that and turn it into a different type of chemical energy, right? And in this case, that would be ATP. We can use that to make ATP, and we'll have animals. They can take that chemical energy. They can make it a different type of chemical energy, right? They could store that as glycogen, and they can break down that glycogen and turn it into ATP. And we're going to lose heat here. We're going to lose energy as heat here. We're going to lose energy as heat here. All these processes. If you're an animal, what do you do with that chemical energy? What kind of energy can you convert it to? What was that? Mechanical energy. Yeah, good. Right? You all look like you're writing right now. So you're turning that ATP energy that you got from plants, or you got from animals that got from plants, got it from plants that got it from the sun, into mechanical energy to write. Okay, so we've got all these types of energy that we can. It's mostly going to have conversions into chemical energy, but you know you'll convert some of it into mechanical energy at times as well. You could imagine. You could also take this and you could turn, either directly or using your ATP, you could turn this into electrochemical gradient energy, right? That's a form of energy. And you're actually spending 25% of your energy right now doing that, okay? That's your sodium potassium ATPase. Questions on that kind of process overall? Okay, I'm going to do a quick aside here because I've been reading papers as part of my research on nuclear organization. And there's some really cool things involved with that. So, we talked about the nucleosomes, but talking about regions of DNA that aren't even on the same chromosome, often they are, but how they get in the nucleus, they end up with active regions in the same. So you've got your whole chromosome right here, and you've got pieces of it that aren't near each other, and those end up coming together in the nucleus into a region this region's activated, like spatial region. This region down here is not active, and they get collected there. You've got proteins that time will cross and pinch them, and things like that. Really cool stuff. And I was thinking about that, and I watch a lot of cosmology videos on YouTube, which is watching space and science and Fermi paradox and things like that. And I can't, from a scientist's perspective, think of anything when I read these sorts of things. I can't think of anything that is more complex than life. Right? There's just nothing else out there that we know of. So I think it's just really, really fascinating the levels of complexity you can get out of these systems. Anyway, I think it's super cool. You guys are just waiting to get out of here and go for Thanksgiving break. Maybe not exactly. Okay, so we'll just do one more thing in general here. I want to talk about catabolism and anabolism. So these are general concepts. So catabolism, so we can think about metabolism. Let's do that. Metabolism is made up of catabolism and anabolism. When we think about this is how we're going to break those processes down. Okay, and so we've got catabolism and anabolism, and what essentially anabolism is going to be building things up, and catabolism is breaking things down, but we're going to have some more specific characteristics we want to know. So catabolism. All right, so complex molecules are made into simpler molecules, right? We're breaking things down. Also in the process of catabolism. So I've got these complex molecules being made up into less complex or simpler molecules. What's going on here? Let's look at we got NAD plus and NADP plus and FAD. The NAD plus becomes NADH, the NADP becomes NADPH, and the FAD becomes FADH2. So we've got this process here, and these guys come in and they go from things like NAD plus to NADH. What's happening to our energy cofactors? These are energy cofactors. They're being reduced, right. So what must be happening to these other molecules? They're being oxidized, right. So the complex molecules being broken down are oxidized, or the atoms are oxidized. And energy cofactors will be reduced. Okay, and that's the point. You're going to take this type of chemical energy and you're going to turn it into this kind of chemical energy, and then you're going to do something with it. Okay, you've got enzyme cofactor energy that's going to turn into, that you're going to use for things. Anabolism is the opposite. Okay, building molecules up. All right, and so in this process, you're going to use those energy molecules. So complex molecules are made. The energy to make them comes from the energy cofactors being oxidized. And those complex molecules being built up are being reduced. Questions? Okay, I think I'm going to stop there for the day. It pains me to stop 10 minutes early here, but do it for Thanksgiving. Enjoy your break. If anybody has any questions or if they want to come see me this afternoon, please let me know. And you guys have a great break.